All right, let's start. Okay. Good morning, afternoon, evening. Hola, hello, bonjour, hola, ni hao, konnichiwa, 안녕하세요, and hello all. And welcome and thank you, SynBYSS people, for joining us the 53rd seminar, and I call it University of South Florida Day or USF Day or NSF Day because of our, pioneer speaker today. This week, September 27, EBRC, a profit or government organization focusing on synthetic biology research and collaboration, and also the main supporter of this SynBYSS seminar series, released the Engineering Biology for Climate and Sustainability, a research roadmap for a cleaner future. This is a year-long collaborative effort of more than 90 people. I just wanted to thank all for this amazing contribution to solving climate crisis or emergency. The almost 200-page document is freely available at ebrc.org for anyone interested in this topic, and freely, you could have access to that document. I would say one disease would kill one person. I had been dying for many years and miraculously recovered from multiple diseases. However, we saw one virus, COVID virus, has killed millions of people worldwide. Similarly, or even worse, climate crisis would eliminate all human species if we do not act immediately. We must solve this global problem, otherwise there would be no future for my daughter and all of us. Despite of this, sad kind of reality, I'm very optimistic because of young scientists and engineers who wanted to solve this problem. So thank all, especially young people and researchers, to tackle this problem, together. Okay, it's my tremendous honor to introduce very briefly our pioneer speaker, Professor Ramon Gonzalez. We all know him, so he does not need any introduction. He is currently a professor at University of South Florida. He is the editor-in-chief of Journal of Industrial Microbiology and Biotechnology, JIMB, and now on NSF MCB program director. He was also a DOE Alpha-E program manager in the past. Through these program director roles for many, many years, he has contributed to the community in a big way, leading the field. Therefore, it is my great honor and pleasure to have Ramon today. And Ramon, the virtual podium is all yours. And thank you so much for your time and contribution to the community. We absolutely thank you for that.
Thank you for taking the lead on this initiative, and for many others as well that you're involved in. I think this is especially rewarding and especially important because it's focusing on young people like Avinash. So I think, as you have stated in the past, that they are the future. So we better take care of the future today. And that's in part the small contribution we're making by holding these type of events. So I thought a little bit about what to say, right? You're going to introduce Avinash, and I am very impressed by what he has already achieved and what he's working on, but he's going to tell us about that. So I'm going to give a couple of remarks that are essentially directed to people like him, the younger generation. So I'm going to share a couple of slides here. So give me 1 second and we will get it done. You can probably see my slide here.
Yes.
So we are living in an era that people doing research in the space, we do research, we are privileged to live in this era because of the contributions and the potential impact of what we do. I put together this slide probably like almost 10 years ago. And back then it was a little bit more futuristic. I think today is a reality. No one would kind of challenge that probably. And the whole point is that these general space of synthetic biology, engineering biology, leading to biomanufacturing and the bioeconomy has the potential and is already becoming the next or the current technology and revolution with impacts that can be seen everywhere in our daily life, from, of course, the typical impact on health, but climate, energy, materials. Once again, Avinash is going to explore a very interesting angle of all of these and very unique. But young people like those who listen in and like our speaker are, I think, privileged to live in this era in which not only they can do outstanding science, but also the impact of that science is going to be huge. So again, I think we all can put together a slide like this. It's a matter of preference. Let me just, my pointer here. It's a matter of preference of what you define as big things that have happened along the years, right? My biases went into these 5 here, but I'm sure I could have chosen others. But bottom line is, this is our era. This is the era of synthetic biology, engineering biology, and biomanufacturing. There is absolutely no doubt on that. I typically, when I have these conversations, I add at the bottom here a few bullet points that make the use of biology for biomanufacturing purposes very unique. And I'm not going to dwell too much on these, but if anyone is interested later on, you can look at something we wrote in Science about 5 years ago. I think is still very much current today. But in essence, there's a huge advantage of using biology as a technology for doing all kinds of things. And that could be the simplicity, the environmental, low environmental footprint, the ability to operate at smaller scale. And because of that, as a consequence of that, you have a bunch of other advantages. And then especially the last one here, last bullet point I wanted to emphasize, in the sense of how biological technologies created through synthetic biology can contribute significantly to faster innovation, adoption of more players, meaning participation everywhere by almost everyone, which then leads to a democratization of whatever industry we are impacting with that. That being the energy and fuels and chemicals, that being climate protection. So that is going to be affordable to pretty much all if we do it through biology. I know that for you guys, I'm talking about and kind of addressing now the younger audience, right? For you at the beginning, it's kind of hard to try to think about big picture and these kind of things, but I strongly encourage you to do so. It's very rewarding to think big picture beyond the science you're doing. What is the impact of that? So I encourage you to read a few general big picture reports there in the space. One is and this one is getting a little bit older. All these things are get older very quickly. But this one is about the US bioeconomy, is the broader thing you can think about, is the entire bioeconomy. This one, and there are more reports, right? This one is produced by the McKinsey Global Institute, and I found this one also very, very useful. This is all big picture, and not your lab, your science, your projects, but the impact that it could have. And Tessa just mentioned the last one that came out from EBRC. There's a number of reports produced by EBRC, and they will continue to produce reports. So this is very, very useful for you to do. So I am kind of making a call here for you to look into this literature, because I know you're going to generate great stuff and contribute great stuff in the scientific literature, but it's very important to be aware of these aspects as well. And the very last thing I wanted to tell you was, when you think about these, and then you think about the work that you do, I would also encourage you as a young scientist to, the way we say here in US, swing for the fences. So look for the moonshots, do that type of stuff. Don't settle for more conventional or traditional things. And while I was thinking about mentioning that, I remember that about a decade ago again, when I was at RPI, we had these type of diagrams in which we will try to define transformational and disruptive type of innovations. And I put together my own one for lighting technologies, let's say a light bulb, right? The diagram in essence tells you how performance, don't think about costs if you want for a second, just think about performance, how that performance relates, how it changes over time for a new technology. And okay, the original one here is the incandescent lamp, right? And there was a learning curve for that. It was created, and over time, performance improve and costs decrease, right? If you come into this area, and this is true for metabolic engineering, synthetic biology, anything else, there's going to be something established. Try to set yourself completely apart from that, creating your own learning curve, your own kind of transformational and disruptive innovation. And in this case, for these technologies, there was a lot of things that were transformational. There were a huge deviation. Now, a lot of these things by definition are not going to completely pan out. And here you have a couple of examples of alternative that, yes, they are actually in use, they are happening, but they couldn't take over. And the one that I claim that is or will or has taken over is LED type of technologies. And so keep in mind that you can come up with these transformational, really creative stuff, and that might not become mainstream ever, but if you don't come up with them, they will never become the established technology or state of the art and so on. Then for some of you might be thinking, well, I want to become a faculty member, and what am I going to do with all of these, right? This is not material for science or nature or whatever. I want to challenge that notion right with this example here, because probably most of you remember that this down here was worth a Nobel Prize in physics. And again, Nobel Prize in physics recognize this disruptive type of innovation. I'm talking about the one in 2014, and it was awarded for the invention. I mean, this is the language taken from the award itself for the invention of efficient blue light emitting diodes, which then enable, what we know as LEDs and with all the implications and so on. So don't settle on the established path. Try to do something very, very different. And that is going to be very rewarding, not just because of the impact and what I was showing in the previous slide, but scientifically speaking, you probably agree with me that this prize is above anything else that we can think about in terms of recognition from the community. So I just wanted to mention essentially those two messages with you. Number one, this is a special time for someone like you, a young scientist who is starting or about to start or has recently started their careers because of the great opportunities that synthetic biology, engineering biology, and biomanufacturing presents. So go all in. This is the time. You're lucky to be in that position. And number two, don't settle for anything that is common, conventional wisdom, let's say. Go for the disruptive, transformational type of research, and that's going to be very, very rewarding. So that was essentially my my 2 bullet points. I promise I didn't want to talk about metabolic engineering, pathways, or any of that. And in any case, if I would have talked about that, Avinash would have made it look really boring, because the type of stuff that he does, again, Tesak, you're going to introduce him. I don't say anything, but the type of stuff that you are doing and are planning to do is outstanding. And to be frank, is right on that, on the categories I was describing, or under those 2 bullet points. Thank you both, then, and back to you, Tesak.
Sure. Wonderful. So you know what? I absolutely 100% agree with you. I mean, we want to make big impact, and big impact coming from sometimes taking risk, bold idea. And also, I also echo what you said in a little bit different way. And many people probably try to get a job or stay in the academia by getting tenure. But this job, academic job, this is not the 5-year, 100-meter sprint race. This is we're talking about 30 years, 40 years of long-term career. So without big picture and big goal, sometimes ambitious goal, without that, I mean, what would be the joy of doing research? Sometimes research is very challenging, but we will be always excited by big goal and challenging goal. So absolutely, you are right, and I love it. Thank you, Ramon.
You're welcome.
Okay, thank you. So now, the main speaker of today with a longer introduction, Dr. Avinash Manjula-Basavanna obtained his PhD at Nehru Center for Advanced Scientific Research, India. He then moved to Harvard University at a Wyss Institute postdoctoral fellow to work on engineered living materials, wherein he genetically engineered microbes to produce sustainable materials with integrated life-like properties. And in fact, the EBRC released one roadmap document, as Ramon already introduced. Recently, he has moved to Northeastern University along with his PI from Harvard and worked at MIT as a research affiliate. He has made pioneering contribution to the emerging field of engineered living materials, and his research interests are at the interface of synthetic biology, materials engineering, nanotechnology, protein engineering, additive manufacturing, and sustainability. In 2020, he was featured as one of 35 innovators under 35 by Technology Review again, MIT. He was the grand prize winner of NSF Big Idea competition and also recognized with Deep Tech Pioneer and Ghanaian Young Technological Innovation Award. And that's another reason I call today NSF Day, because both of speaker today, has common thing, that's NSF. He was also selected for the Innovation Scholars in Residence program by the President of India and Harvard Innovation Labs Venture Incubation Program. His research work has been highlighted by several global media outlets, such as the New York Times, the Washington Post, New Scientist, to name a few. He is truly a rising star, and I feel like he already risen. And it's my great pleasure to have him today. Avinash, thank you so much for your time today, and please take it away. Thank you.
I hope you all can see my slides.
Yes.
Okay. Thank you, Professor Moon, for your kind and generous words. It has been a privilege to connect with the SynBio community and also to share this virtual stage with Professor Ramon. And all of this, I don't think, would have been possible if not for SynBYSS and the efforts which Professor Moon has put in creating this. Thank you, Professor Moon, once again. And so I've been working at the interface of synthetic biology and materials engineering, which has led to a new realm known as engineered living materials. So I started working on engineered living materials when I moved to Harvard University's Wyss Institute way back in 2017. And subsequently, I've continued my work at MIT and Northeastern University. So, so when we look around, we can find remarkable human-made materials that have enabled our way to day-to-day activities. And also it is important to realize that it has also shaped the course of human evolution. But at times we don't take into account that the kind of environmental impact that some of these synthetic materials have been making in changing our ecosystems, which I think is mainly because of the unsustainable make, use, dispose practices. Adding further, we should also take account of the fact that human-made materials are fabricated by what are known as heat beat treat strategies, mainly due to their energy-intensive or harsh temperature, harsh chemical treatments, or high temperature, high pressure processes. But in contrast to this, biological materials are produced at ambient conditions from abundantly available benign components. These differences between the human-made materials and the biological materials inspired me to work on bio-inspired materials engineering during my PhD. But by the time I was finishing my PhD, I started to wonder, instead of just taking inspiration from natural systems, why not utilize the manufacturing capability of the living cells to make materials of our choice? And in the past 20 years or so, with the advent of various synthetic biology tools and techniques, living cells have been utilized as factories to produce fuels, chemicals, drugs, fragrance, and even food. But it has been relatively less explored for something like producing functional materials. It's also extremely important for all of us to take note of is the fact that, as per a recent estimate, the human-made synthetic materials outweigh all living biomass. It is quite staggering to think that we have been able to produce materials and we have been utilizing materials on our day-to-day basis, and which has outmassed that of all living biomass. So it's high time that we also need to take these actions much more in a careful manner and also look for a better future for all of us. It is in this regard, few of us are working in this field of engineered living materials, are trying to utilize living cells as factories to produce materials. And our big vision is not just about producing materials, but also integrate the materials with lifelike properties, like that of self-regeneration, self-healing, just like our bones, which can heal the crack, or the cells that is present in our body can regenerate and respond to external stimuli and things like that, can we utilize these kinds of living characteristics in our materials? So this is the vision with which we are working in the field of engineered living materials. With the big vision of the engineered living materials is just like a seed that has all the necessary genetic information stored within to self-replicate, acquire resources from its surroundings, and grow to a gigantic tree. Can we engineer living cells to make macroscopic materials with lifelike properties? In doing so, we believe that we should be able to restore a circular material economy, which is unlike the make, use, dispose linear material economy of the current material that we have. So in doing so, we think that we should be able to contribute towards building a sustainable world. So let's now take a moment to understand how do we make such living materials. For this purpose, we use what are known as biofilms. Biofilms are organized communities of cells that are embedded in an external cellular matrix. And it is this matrix which provides them properties like substrate adhesion, protection, as well as microbial persistence. So we in our lab have developed a technique to engineer this extracellular matrix, which we call biofilm integrated nanofiber display technique. And so when we talk about extracellular matrix, it could be made up of either proteins, polysaccharides, and nucleic acids. The fact that the nucleic acid-based materials have limited properties, whereas the polysaccharides have limited control in terms of genetic modification, whereas the proteins are quite versatile and can be easily modified genetically and can produce different kinds of material properties, protein-based ECMs have become the point of interest in the ALM community. So we specifically make use of a particular biofilm, which we call curly biofilms. These are the biofilms which have an extracellular matrix made up of curly nanofibers, which are typically have a diameter of around 4 nanometers. And these curly nanofibers are produced, are made from a protein called CSGA. And these curly nanofibers have very robust characteristics as they're resistant to heat, solvents, denaturants, detergents. So by using this bind technique, we can genetically engineer this E. coli such that we can fuse any desired peptide or protein to the CSGA building block, which forms this curly nanofiber. So, so these engineered proteins, once it is produced inside the E. coli, it will be secreted out. And once it secretes out, it can self-assemble to form this protein nanofibers, wherein which you have this engineered peptide or the desired peptide or protein will be displayed on its periphery to get this functional curly nanofibers. So all in all, we want to utilize this as a one-stop microbial platform to produce living functional materials. So these curly nanofibers are produced from a well-established and sophisticated curly secretion machinery, which has 7 genes, which regulates the production as well as the secretion and their self-assembly in the extracellular space. So this is a video where you can see the schematic shown in blue is the CSGA, which is linked with a linker shown in yellow to a desired peptide or the bioactive peptide shown in green. So once this protein is produced inside the bacteria, it will be in an unfolded state. And once it secretes out to the extracellular space, it will fold to its native state. It will be folding on top of a nucleator protein called CSGB. And this CSGA, which folds to its native state, which is a beta helical in nature, whereas this functional peptide or the protein that we have fused will be displayed on its periphery. As more and more of these monomers gets secreted into the extracellular space, they self-assemble on top of each other, such that the functional peptide will be displayed on its periphery. And ultimately, it results in the formation of a functional curly nanofiber, like you see it here. And once, if you try to visualize this in a scanning electron microscope, these are the engineered bacteria which are surrounded by this functional curly nanofiber mesh, which is what we utilize for getting the functional nanomaterials. So some of the earlier examples of this field of engineered living materials showcase that it can be used for binding to synthetic surfaces like that of stainless steel, or bind nanoparticles like gold or silver, or even we can utilize for displaying proteins or enzymes in a 2-step mechanism. So when I joined Joshi Lab in 2017, my focus was to push this field into new directions and produce some materials which can have real-life applications, and to integrate lifelike characteristics, and to have some advanced functionalities which has not been shown before. So with that objective in mind, I have been able to push this field into totally new terrains, like by developing a transient living material, structural living material, stiff living material, hybrid living material, and sequestration living material. Let me first start with the transient living material. So the motivation for this particular work came from the plastic pollution, which is a global problem. As you might be aware, as of now, 8 billion tons of plastic has been produced, and only about 10% of the plastic gets recycled. Add to that, you have the microplastics, which has contaminated almost every part of the globe, including human gut and lungs. And there could be as many as 100,000 microplastics in human guts and lungs. So clearly, plastic is not just an environmental issue, but also an upcoming global health problem. So one way to circumvent this plastic problem is to replace, at least wherever feasible, with bioplastics or biodegradable alternatives. So this one particular area where bioplastics could be utilized is something like packaging. Packaging contributes to about 1/3 of the entire plastic market, and a typical lifespan of a packaging material is only about 1 to 2 years. So when you have a lifespan requirement of 1 to 2 years, there is no need for us to think of non-biodegradable plastics and which can last for hundreds of years for an application which is required only for 1 to 2 years. So, but unfortunately, what has happened here is that only about 0.3% of the plastic market comprises of biodegradable bioplastics. And if you think that you can utilize land-based sources like starch or corn to produce these bioplastics, one should also keep in mind that the available arable land is limited and it will not be able to cope up with the global demand. It is in this scenario, microbial production of biodegradable bioplastics becomes a promising approach. To this end, we have developed a transient living material, a material which can be used only for temporary basis, and that is what we call aquaplastic. Aquaplastic is the world's first bioplastic produced entirely from the engineered microbial biofilms. And it has several unique characteristics, being that it is water processable, like that of aqua molding, aqua welding, as well as aqua healing. Unlike petrochemical-based plastics, which involves thermal molding, these aquaplastics are molded at ambient conditions by a process known as aqua molding. So what we essentially do here is that we have this engineered E. coli, which produces these functional curly nanofibers, and you can just filter it out on a filter membrane to produce this aquagels by a simple treatment. So once you have this aquagels, you can cast it on a 2-dimensional surface to get a 2-dimensional aquaplastic films, or you can use a 3-dimensional sacrificial mold to get 3-dimensional objects. So this process of fabricating aquaplastic from the aquagel is what we call aqua molding. So the plastic nature of the aquaplastic came from the tensile test. As you can see, the plastic deformation from this tensile stress strain curves. And also the thing that we could see was that the Young's modulus was about 1 gigapascals and the tensile strength was about 20 to 30 megapascals. And these values are comparable to that of the petrochemical-based plastics as well as the bioplastics. But aquaplastic is biodegradable, unlike the petrochemical-based plastics. It can biodegrade up to 90 to 95% in just 45 days, whereas the gold standard for biodegradation, nanocellulose, could degrade only about 85 to 90%. So aquaplastic clearly is a biodegradable bioplastic, which is having interesting properties. When we're talking about properties, like I said, it also has something called aqua healing. So what does it mean is that if there are scratches or cuts in the aquaplastic films, then you can just use water to heal this scratches and cuts. Or else you can even push this further, like in the sense that if you have 2 pieces of aquaplastic, then you can also use water as a glue to weld them. You can weld them laterally, or you can also weld them orthogonally. So these welded structures are also quite robust, as you can see from the tensile test. As a fun experiment, we wanted to utilize this welding capability to build something 3-dimensional structure. So we made these panels and we welded them just by using water. We could ultimately build a 3-dimensional structure, which we call Aqua House. And this is still intact even after 2 years of its construction. Keep in mind that this is being welded just by using water and nothing else. This attracted a lot of media attention, and especially from Washington Post and many other news media outlets. So if you're concerned about the susceptibility of these, water susceptibility of these aquaplastic, then you can also modulate their water susceptibility by introducing some patterns on their surface, like that of micro and nano surface patterns, by using which we can push their contact angle. Typically, aquaplastic has a water contact angle of around 58 degrees, which is an indication that the material is hydrophilic. By imprinting this material on patterned surfaces, you can get this water contact angle to about 117, which is hydrophobic in nature. And aquaplastic is made from curly fibers, which I said are robust. So then we wanted to test how robust are these materials made from these curly nanofibers. For this purpose, we put this aquaplastic into various chemicals, like there are organic solvents like hexane or even the corrosive chloroform, which dissolves many plastics. But the aquaplastic was intact in these solvents even after 24-hour exposure. And also they were quite stable in concentrated sulfuric acid and sodium hydroxide. Although the material, the aquaplastic swells, but the material was still something which did not degrade. So if you can couple these resistive properties of these aquaplastic with their ability to bind to surfaces which curly fibers are known to have, so then it can be utilized as a protective coating on different surfaces. And for this purpose, we have shown that it can be used as a protective coating on leather or plywood, mobile phone touch screens, and then also automobile exteriors, as well as even copper wires, which can be used as an insulation material. So clearly, some of the examples which I showed till now was not something which can replace the aquaplastic, but the motivation was to address towards that plastic problem. What I'm currently doing is working on the second generation of aquaplastic, and these are some of the prototypes which are made. What you have is a material which feels just like a plastic wrap, which is commercially available, ultimately we want to utilize these materials for packaging applications. Some of these works are still ongoing, and I hope to put them into public domain or publish within the end of this year. So, so let me now switch gears to another work which I have done, which is called structural living material. So what I'm trying to emphasize here is the fact that if you're trying to build a 3D structure, starting from, let's say, a microbe, like the one which I've showed in the previous aquaplastic work, you start with a protein like the CSGA, and then it can self-assemble to form these fibers or aggregates and form this aquagel and make these films, and you weld them and to make this three-dimensional structure. But what if you want to build a structure which is a complex 3D architecture? In such cases, this clearly cannot be utilized for such applications. And in that scenario, one could think of using is a 3D printer or a 3D printing technique. To do that, you need a ink or a bio ink, or in recent years, there is an emergence of a new type of ink, which is called living ink. So what living ink is basically an ink, which is bio ink, which is used for 3D printing microbes. So when you look critically at this living ink, so what these existing reports do is they make use of a bio ink, which is used in tissue engineering community instead of printing it with mammalian cells, they replace it with microbes, and they use it for 3D printing. And this is what is called living ink. So what we wanted to do was to produce this ink entirely from the microbe and not use just some of the existing bio inks. So in doing so, we should be able to produce an ink entirely from the engineered microbe, and we can use for printing. And if you can introduce additional engineered microbes into these ink, then we can get functional living materials. So to do this, we took inspiration from fibrin. Fibrin is a protein which is involved in blood clotting. During blood clot, there are several protein domains, like there are alpha and gamma, which interacts together, which cross-links and leads to fibrillation. So these alpha gamma interactions are also known as knob and hole interactions. So what we have done is we took this alpha and gamma domains from this fibrin and then genetically integrated that into CSGA in two constructs. In one construct, what we have done, we have fused this alpha domain to CSGA. In another construct, we have fused this gamma domain to CSGA. So once these engineered proteins are produced and secreted, so they self-assemble. CSGA will stack on top of each other by displaying the alpha and gamma domains on its periphery. And if these alpha and gamma domains are still bioactive, then they should be able to cross-link. And if they cross-link, thereby we might be able to modulate the rheological characteristics of the ink and thereby get a favorable ink which can be utilized for 3D printing. The overall scheme is something like this. You start with this engineered bacteria, two engineered bacteria, and then you co-culture them and you express them so that these engineered fibers will cross-link. And if this cross-linking strategy has worked, so then you have an ink which is good enough for it to be utilized for 3D printing. And by introducing additional engineered microbes, we should be able to get additional living material characteristics. So this was a quite challenging problem. We did struggle with this for a couple of years, and then ultimately with this design, what we have shown is that in terms of rheological characteristics, storage modulus of the cross-linked ink was much more higher than that of the non-cross-linked or just the alpha or the gamma constructs. The viscosity was also significantly higher. And as you can see that the viscosity of these materials decreases with increasing shear rate, which is an indication that the material is shear thinning, and thereby it can be used for extrusion-based 3D printing. The shear modulus was also higher, as well as the yield stress was also higher because of this protein-protein cross-linking. The printed structures were also having better shape integrity, as their printed diameters, filament diameters was like 500 micrometer for the cross-linked ones, and the non-cross-linked ones were about 1000 or 1500 micrometers. An ultimate test for the shape fidelity of these inks came from the filament collapse test. So what we have done here is that we have these printed pillar structures. So each pillar is having a distance from its neighbor, like there are 1 millimeter, 2 millimeter, 4 millimeter, 8 and 16 millimeter. So now what we do is we extrude this ink on top of these pillars, such that if the ink is having good shape integrity, shape fidelity, then it should be able to form a bridge across these pillars. As you can see here, this is the ink obtained from alpha gamma cross-linked ones. And as a result, it was able to even form bridge when the gaps are of like even 16 millimeter. In case of the non-cross-linked ones, the alpha or the gamma, they could not form any bridge beyond 4 millimeter gaps. This was clearly a good reflection of the fact that we can engineer microbes to produce materials and modulate their mechanical properties to get a material which is, in this case, a bio-ink, which is 3D printable. So using this, we try to print wide variety of 3D structures, different shapes and patterns. And ultimately, we could introduce additional engineered living materials in these printed structures. In this case, we have developed a therapeutic living material. So we have built in this genetic circuit in which you can induce with IPTG to produce an anti-cancer drug, azurin, as can be seen from this gel images, that only in the presence of IPTG, it produces this anti-cancer drug and not in the absence of IPTG. So on demand, you can produce this anti-cancer drug in these printed living structures. In another case, for some applications, let's say you don't want to use the living cells, or somehow you want to control the amount of living cells present in your material, printed structure. For that scenario, we have developed a regulatable living material where you can also induce this with IPTG to produce a toxin, a MASF, and this toxin kills the cells, and thereby we can control the amount of cells present in the material. So, so far I have given you examples where I have engineered the cells to produce an extracellular matrix, which is in the form of functional curly nanofiber to produce the functional material. So one of the things which I started to wonder was, why not use the living cell itself as a material? As you all know, living cell is hydrated and soft in nature. And without having any of these extracellular matrix, what can you do with that? This was a tricky kind of an idea, and nobody believed that something will come out of this. But I was persistent with this idea and then kept on trying and ultimately came up with a simple design. What I have done is that, so any typical microbe, maybe in this case, it's an E. coli, you just culture that, okay? And then you pelletize it, you wash off, remove all the nutrients, and then you cast it into a mold, and then you dry it out. So you dry it out such that you get a material which is cohesive, glassy, and it is quite stiff. So how does this happen? The trick here is the kind of mold or the casting that you use. When I started this work, so what happened was, if you cast this pellet and put it on a glass surface, so what you would get is something like this, where you have a flaky material which has a lot of cracks and are not at all cohesive and nothing interesting apart from maybe some kind of artistic sense to it. But I was persistent with this idea and then went on changing different parameters like substrates, temperature, and so forth. And ultimately, after playing around with these with different results that I got, made some intelligent modifications and ultimately gave material, which you see in the bottom right, which is glassy, cohesive, non-crack, cracked material. And this is something which is produced entirely from just the E. coli. So people have been working with bacteria or E. coli and many of these microbes for centuries. But to think that you can make a material just by using cell was something quite amazing for me at that point of time, at least. So what I have done here is, this is a material which I call stiff living material, produced entirely from the E. coli. And to test the applicability for different kinds of microbes, I have used a gram-positive bacteria, Lactobacillus. But in this case, the material, although was having a kind of a shiny, glassy nature to it, but the surface was corrugated and it was assembling into patchy domains, and not that it was making a fully intact material. In case of yeast, the material was intact, but the material was not glossy, and it had an air pocket at the core, and the top and the bottom layers had a lot of cracks, but although the material was still holding onto each other. So since you have started this material fabrication from using living cells, the other question was to see if any of the cells are alive in this material. And to our surprise, what we found was that nearly 1/3 of the cells are alive in E. coli-based SLM, as well as that of in yeast-based SLM. But in case of Lactobacillus, all the cells were dead. It is still surprising for us to see that or understand how these cells are able to survive, and that to 1/3 of the cells are being alive within this material. So since this is produced from cells, so you would naturally expect that the material is amorphous, as you can see from the X-ray studies. It looks a bit of transparent in nature, but by the absorption spectra, what we find is that the transparency is less than 10% across the entire visible range. By using ion notation, I was telling you that the material is stiff. So this was quite astonishing to see that the stiffness is about like 5 to 10 gigapascals. And their hardness is about 100 to 200 megapascals. These values are insanely high, and to expect that you can get, and just by using living cells, is still something which I'm finding it difficult to comprehend. Since you have living cells within this material, then the idea is like, can you take a piece of this material and can it regenerate itself, and which can be utilized for producing successive generation. So, so I have taken a small piece of this material, put it into media, it regenerates, you do the same protocol, and then you produce these successive generations. And these successive generation materials are also having the similar living cells characteristics, viable cells within these materials. Their mechanical stiffness is also in the same range. So since you have living cells within your material, then the obvious question is, how long can these cells survive, right? So, so what we have found is that by time dependency of your analysis, these cells are able to survive, at least in these E. coli-based SLMs, up to over a month's time. So you should keep in mind that you don't have any nutrient media, and you're not replenishing any medias to it, and and still they're able to survive. In natural settings, if you were to just use some put some bacteria into open air without any nutrients, and they would die out within a couple of hours or within a couple of days. But they are able to survive to up to 1 month within this material. So just to give you a kind of a sense of the mechanical landscape of these stiff living materials, I have plotted Ashby plots here. So what you see here is that the stiff living materials have the like this Young's modulus, their stiffness and the strength, which are comparable to many known structural materials like that of wood, silk, or even plastics, and even many other composites. And all these things are being produced from a cell which is heterogeneous in nature, right? So it has like 50% of protein, like there are dry E. coli cell, and there are so many other components. And in spite of being such a heterogeneous entity, how they are able to self-assemble and to form such cohesive and intact glassy material is still something which we are trying to understand. Nevertheless, but this has been a worthwhile exploration, and I did not listen to naysayers, and I was persistent with this idea, and it's good to see that has reached to a certain level like this. Let me now briefly touch upon two other work. One is on hybrid living materials. So here the idea was to push the living material, engineered living materials context to into a next level. So what we wanted to do here was to use two different bacteria, and it is a case of division of labor. Like if you use one bacteria to do one task and other bacteria for doing other tasks. So how do you get this kind of a material? And for that, what we have done is we came up with a clever design Like I was mentioning about superhydrophobic surfaces, on superhydrophobic surfaces, if you put a droplet of water, it attains a spherical shape because of its high surface tension of the water, and to minimize its surface energy, it attains a spherical shape. So now if you take a media like that of HS media, and within this media, you have, let's say, two different kinds of bacteria. One is gluconoacidobacter and then E. coli. So, and if you put it on this PTFE powder, because of the superhydrophobic nature, this media attains a spherical droplet shape. And within this media, you have these two bacteria. In this media, HS media, the gluconoacidobacter starts proliferating, and it produces a nanocellulose at the air-water interface. And so thereby, if you keep this for like a couple of days, so it produces a nanocellulose-based capsule. And if this capsule, if you put it into another media like LB, where the E. coli cells will proliferate, and thereby you can get the engineered E. coli to produce some functional aspects that you want them to. So this is a capsule produced by this gluconoacidobacter. And as you can see, this is the nanocellulose formed at the capsule shell. And now you put this into another media like LB media, where E. coli can proliferate. And as you can see here, in this case, the E. coli is producing the red fluorescent protein, and thereby you get this red capsule. So now you have this E. coli, which can be genetically engineered, either to display some nanobodies or enzymes, and even to an extent where you can facilitate mineralization. These capsules were soft in nature, and because of this mineralization, you can also make them hard capsules. So now let me briefly touch upon the last work, which I call sequestration living material. So in this, what I have tried to do is to develop an engineered living material to recover some valuable resources, like that of lanthanides, which are used in modern consumer electronics. Because lanthanides are available in limited quantities, it is important that we try to recover and reuse these lanthanides. So we have developed this lanthanide binding engineered living material, which can selectively bind these lanthanides from the waste effluents. And these bound lanthanides can be recovered and can also be, and this engineered living material can also be reused for a couple of cycles. So this can also be customized for sequestering toxins or pathogens, and these are specifically important in the time of the corona pandemic that we have. So, or you can also customize it to sequester a toxin or a carcinogen like that of bisphenol A from the contaminated water. So all in all, in summary, I hope I was able to convey to you that engineered living materials is an emerging and exciting field which integrates synthetic biology with materials engineering. The emphasis is not just about producing materials, sustainable alternatives, which is important. We are going one step further in trying to integrate lifelike properties for creating smart materials. So with my work, I have given you examples of self-regeneration in the form of the stiff living materials, self-healing in the form of aquaplastic, aqua healing, and aqua healing characteristics. Self-regulation and environmental responsiveness, or something which we have shown in the microbial ink work, and the autonomous or the biosynthesis is something which I've shown by the production of biosynthesis, microbial ink, as well as in the form of this capsules, hybrid living capsules that we have produced. In terms of applications, one which I've shown is the sequestration of valuable resources or the toxins, and can also be for the biodegradable bioplastics for packaging applications. So overall, some of my work, specifically in the engineered living materials, has got me the recognition as the world's top 35 innovators under 35 by MIT Technology Review. The idea of and the big vision of engineered living materials proposed by me and my colleagues was given the grand prize winner in the NSF Big Idea competition. And a couple of my work has also featured in global media outlets like that of Washington Post, New York Times, and Smithsonian Magazine. With that, I would like to acknowledge Joshi Group, my collaborators, and the funding agencies. And finally, I'd like to thank you all for your kind attention, and I will be happy to answer any of your questions.
Wonderful, wonderful talk. I have a couple of questions, but before I ask the question, I want to ask Ramon whether you have comment or any challenging question.
Yeah, this is outstanding. I think this is a great example of the opportunities at the interface of biology and other disciplines. I think some people refer to it as convergence type of research because of the convergence of different disciplines. What are the next steps? Where do you take it from here?
So I think it depends on the kind of projects. Like one of the things being Aquaplastic, where I'm seriously focusing on trying to make prototypes which are relevant in the packaging industry. And then in terms of in a bigger picture wise, is that how do you produce these materials in scalable quantities? Like I said, to start with, a couple of years ago, most of the materials were produced in nanometers or micrometer scale. And now I've been able to push this into several centimeter scale. And these are all done with the typical laboratory shake flask settings. But if I switch to bioreactors, and then I can easily scale them up. And then the obvious question is, if you're wanting to have just the material production, it is different. But let's say if you want to integrate life-like characteristics, you want to have living cells in your material. So how do you contain them, right? So what are the kind of biocontainment strategies that you want to use? These are certain questions that one has to look into. But yeah, I think we are just scratching the surface and trying to explore. As of now, we are trying to do what kind of new functionalities, what new things can you do? And then the question is like, how do I take it to the next level in terms of real life applications is what mostly would be focusing on.
Wonderful. Ramon, do you have any other question?
No, thank you.
Okay, so Ramon asked one of my question. Now I only need two questions, but I wanted to address one question from audience. So the question is, are the bacteria aerobic or anaerobic? And the first question. Second question is that how the bacteria survive on the SLMs? And sorry, it might be a silly question or information like that I missed. That's the question.
So yeah, these bacteria that we are mostly working with are aerobic. And then with respect to second question, so the thing is, see, like I said, before I cast this bacteria into this mold to get this stiff living material, so what I have tried to do is that I have washed all the media so that I did not wanted media components to be a factor in trying to make this cohesive material. And for that reason, I've tried to wash off all the nutrient media. So you don't have a media in that material. But what I've seen is that maybe one of the things which I have not presented here because of the time constraints. So what I have done here is, I don't know whether this is visible to you.
Mm-hmm.
Yeah, so what we have done here is that I try to visualize this material under a scanning electron microscope. And when you see from the top surface, so the material that the living cells are all dead. But when you see at the core, you have domains where the cells are tightly packed. And there are regions where the cells are loosely bound. So when you have a structure like this, where we think that some of the cells are able to survive in these loosely bound or tightly bound domains, and they might be getting some kind of resources or nutrients from them, or they might be going to some kind of dormant state. These are certain things which we don't know exactly. And also there could be other desiccation tolerance mechanisms which can produce trihalose and many other components during their drying phase, could also be contributing towards their cell surviving capabilities during this 1 month duration. But yeah, I specifically don't have a concrete answer of what exactly is the reason, but these are some probable reasons that could be helping them.
Okay, wonderful. So I want to ask some question. My quick question is for your biodegradation experiment about biodegradable plastic work.
Yeah.
Did you add some enzyme to specifically to degrade this one, or you just put that in?
We took just the waste from microbes from the waste effluent, and then we exposed them, and over a period of time we see that the microbes were able to degrade these. We did not add any enzymes. Yeah, it is, we wanted to keep it more like a real life scenario, and then the just the microbes which are there in waste effluents. Yeah,
that's wonderful. Then because I mean that is amazing. I believe you mentioned 45 days, right? In that experiment. And the reason I'm asking that question is people talking about biodegradable polymer or plastic, but that might be misleading because in real life in the scenario, I mean, you put everything in the the flask or the tube and then mix with the bacteria. But in real life, the chance of plastic exposed to the bacteria also low. But at the same time, it takes in realistic sense, probably 6 months or something like that, even though those are exposed to some bacteria. So, but this is amazing. 45 days, that condition still degrade almost 100%. That's amazing. So great. My second question is more, you kind of briefly touch about biocontainment, but I wanted to hear more about your perspective, because especially your living material section, that might be one issue, security or safety type of issue at the end of the day.
Yeah, so there could be a couple of ways how you can do that. Like one being that instead of using, although the strains and everything that we work with is all non-pathogenic in nature, even the E. coli that we use is non-pathogenic. So you can switch to some strains which are like even the probiotic strains, or some things which we are already exposed to, like lactobacillus, or many of this yeast and many of these things which we are exposed to, or see them as a non-pathogenic or commensal kind of microbes to start with. And the second thing, in terms of containment, one of the things is that maybe you can think of using synthetic oxotropes or some things which would function, or they are active only under the presence of those conditions. In that way, you can, and also like I said, we can have this suicidal kill switches, like the way I've used in terms of this MASF regulatable living material that I demonstrated. So you can have a couple of these strategies, but depends on which scenario and what is the need, and things like that. See, if you were to use, let's say, many of the applications are in, like, maybe thinking of, let's say, a concrete, right? You want to have a cement or something like that. There you want to have a material, but in that case, what happens is that instead of using just the living cells, you can use some strains which can convert into a spores. So in that way, they are only functional only when you have, let's say, access to nutrients, or maybe in terms of, let's say, water, where there is a crack, then only these spores are being exposed to water or and then only they can start producing a component which can heal those cracks. So there are different kind of strategies that one can use depending on the applications. Yeah.
Okay, wonderful. So the reason I'm asking that question is I'm very interested in biocontainment, and I'm working on multiple projects. So we may talk later, for the collaboration.
Sure.
So wonderful. So I wanted to conclude because I don't see any other question. And also, interestingly, we chat at the beginning of our seminar. Ramon gave nice design of house, old design, and then Avinash basically showed the house exactly similar way Ramon describes.
Yeah. So, as kids, we were like, one of the things that's like Ramon was saying, so I was trying to do was like build these houses from cardboards, and stuff for some school exhibitions and demos and things like that. So I think that's also something we just carried on over here now.
Yeah, of course. So thank you for your inspiring or pioneering design many decades ago, Ramon. So all right. Thank all for joining and staying today. We'll meet again next week on October 6th, Thursday, the same time, the same Zoom link. We'll have Professor Byung-Wan Jo from KAIST and Dr. Witten Schroeder from Penn State University. As usual, the follow informal chat will occur without recording. Please stay here if you are interested in chatting with today's speaker. And I will stop recording. And thank you again. Give me 1 second.